So I would say the most interesting thing about this tech for the scale-up is the fact that the microenvironment of the cells is the same when you scale up. What I mean by that is that if you see a picture of our tech on our website or online, we'll see if we can attach one to this podcast, why not? But the cells are located into very stable and periodically spaced layers in the liquid in 3D. And this microenvironment will be the same when you scale up to 1 liter, 10 liter. You will just have more layers, basically.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Today, we are exploring an exciting innovation. We're looking at the use of acoustic waves in cell manufacturing. And I'm having a conversation with Gabriel Dumy, who is the co-founder and CTO of Kolibri, a biotech company aiming at redefining the mammalian cell bioreactor through the use of acoustic waves to sort, engineer, and grow these high added value cells in the best manner. Stay tuned for an exciting conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Hi, Gabriel. Welcome to the Smart Biotech Scientist podcast.
Hi, David. Thank you for inviting me. How are you doing?
I'm very well, thank you. How are you doing, Gabriel?
Quite well. So here in Paris today, it's rainy, but very happy to be here and to have these conversations.
Cool. It's so good to have you on the show today. Gabriel, share something that you believe about bioprocess development that most people disagree with.
Probably it will be the importance of the use of adherent cells for bioproduction. You have a general trend, which is to switch from adherent to suspension as you scale your processes up and up and up. And of course, it makes sense because suspension microorganisms can be cultivated at much higher densities, a much better optimization of surfaces, if you want. But one has to remember that all the processes are usually developed in adherent strains, whether it's in academic labs or on very small scales, such as Petri dishes. And you have to make a switch at some point between the two, which is not always trivial. And so most people will only swear by suspension. Only a few years ago, in viral vector bioproduction, you still had a lot of adherent, whether it was on microcarriers, cell factories, roller bottles, and a lot of people are still very used to produce with these strains. So I know a lot of people will disagree on stating that adjuvant still has a role to play in modern bioproduction, but that's something I believe, and that's something that we try to address by designing our bioreactors at Kolibri.
Exciting. So, Gabriel, before we jump into our topic about using acoustic waves in cell manufacturing, let's talk about yourself for a second, because I'm curious, what led you to start in biotech, and how did you end up now in your role as a CTO?
I've always been fascinated by biology and by science in general. When I was a kid, I was reading all I could find on science in itself, and I was taking apart appliances in my home to understand how they were working. And so I have this deep connection to physics and to hardware development, with a biology twist. So I've always worked in biology labs, whether it was as an intern, as a student, then as a PhD candidate, building hardware, experimental setups, early products for my fellow biologists. So it was electric fields to align pneumatic cells or to stimulate cardiac cells, magnetic fields to align zebrafish's embryo under the microscope so we can actually image them better, acoustic waves, we will come to that, to sort, to grow, to purify yeast, bacteria, algae, and mammalian cells. So I've always had this very profound interest in mixing advanced and fun physics or chemistry with a biology application. And so I've done that for more than a decade now in academic, industrial settings, both in France and abroad, in the Cambridge, US, Cambridge, UK, Australia, you name it. And so I really like when I can build a physical product, a machine, basically, whether it's a machine that will perform experiments or a machine that will produce something in the lab, and to have it applied to microorganism manipulation. So as I said, in the past, I've done sorting, I've done purification, I've done growth. And in the past, literally 5 years, I've had a much more precise focus on mammalian cells. So how to improve cell manufacturing or their byproduct manufacturing by either manipulating these cells with electromagnetic or acoustic waves, or by building better products to help the industry face the challenges that are inherent to mammalian cell bioproduction.
So you have seen various parts of the industry, both in academia and in the industry. So I would like to know, Gabriel, what do you think are the main challenges that biotech scientists face in modern bioproduction?
So I would say that you have 3 of them. You have the fact that a lot of the bioproduction instruments are optimized for prokaryotic microorganisms, whether it's algae, bacteria, or yeast, liquid living microorganisms. So yeast, bacteria, algae, all of these things are fine, living in puddles, being, stressed all day by temperature, pH, or mechanical changes. So they are especially suited to very large culture vats where you stir, where you bubble, the required nutriments and gases. When you want to use these machines for mammalian, it works, but mammals are a huge colony of cells. We are not living in a puddle. And so when you separate your cells into individually suspended cells in a liquid, it works, but it doesn't work as well as the prokaryotic does, basically. And so you end up with a machine that you know how to operate, that exists, but that do not perform as well as it does with yeast or bacteria because of the nature of the microorganism that you want to exploit. So that's the first point to me, a gap in productivity so far for modern mammalian cell-based bioproduction. The second one is how to effectively engineer your production units. So today we have very good production strains of E. coli, of Saccharomyces cerevisiae, for instance, that we understand quite well. We have not deciphered every pathway on the biologics that we want to produce with mammalian cells. So monoclonal antibodies, viral vectors. Today it's still not clear how an AAV, for instance, is packaged inside the human cells, how the different proteins VP1, 2, and 3 assembled to form the capsids. And so it's quite hard to engineer the cell the right way to have it produce what you want in the best fashion. And so I would say that's the second point, how to engineer the cells so that they produce better. So it entices modifying the cells with the right DNA or RNA, and how to bring the RNA to the cell. So how to transfect your production unit in the best fashion. That's still the highest cost, I would say, in gene therapy manufacturing, the transfection regions. So how to bring the recipe inside the production unit. So it has a bit to do with the bioreactor itself, but it's mostly the cell biology itself that is still not totally mastered. And the last one, of course, is the fact that you work with human material, that you want to produce a drug, so a pharmaceutical validated ingredient. And so you have huge costs involved. Bioproduction run, a large one for gene therapy, can cost up to $1 million and that's only cost of goods. And so, of course, when you have such stakes, you are not enticed to look for, rupture innovation that can drive down the cost like crazy, because you have a lot of regulation in place, because of all of the issues there has been in clinical trials for the past 30 years, because you want a viral vector that's non-replication capable and so on. And so the fact that you work with human material, pharmaceutical, and with a lot of money, basically, makes it a very conservative industry. And so it's hard to have solutions that could efficiently address the two first points, basically. That's the situation that I've seen today working in human cell-based bioproduction. Hopefully it will change in the coming years, but it is a conservative pace, let's say.
Yeah, I totally agree. We work in a conservative industry. I have been in the business for many years, and that's totally also what I see. I cannot agree more that we are in a conservative industry. But you mentioned the word cost, and you and your team started to develop bioreactors using acoustic waves, particularly in the context of gene therapies. So can you explain us what inspired you to do that in gene therapy?
The concept was to build, in one sentence, a bioreactor that would be designed for human cells, for mammalian. So not starting from existing, directly designing something adapted to the growth and engineering of mammalian cells. And back at the time when we started, late 2020, COVID went over the planet. Bioproduction, especially of biologics, started to gain traction. And so we did talk with a lot of people about how mammalian cells were used and were produced in the industry, especially the pharmaceutical industry. And so we talked with one of the founders of Cellectis, who pointed us towards the fact that Cellectis are modifying their cells. They need lentiviral vectors. And at the time, they had a 2-year wait list to get their vectors because of the capacity crunch that the industry had. Basically, even if you had top money, it was very hard to find the capacity to produce your viral vectors if you need them. And so we discovered at this time, so give or take 3 years ago, the field of viral vector bioproduction. And it's actually very interesting because it involves a lot of painful steps. You need to amplify human cells with all the problems that it encapsulates. Then you need to transfect them, give them the recipe to produce the right gene therapy vector. And then you need to harvest and purify your vectors in the best fashion. And so it proposed to us a problem that was matching quite well what we thought our acoustic technology could do. And so that's why we delved into it, because we found a technical problem that was matching very well the proposition we had, and because the market was really begging for better capacities, still is today, even if the capacities have improved, notably through the use of suspension bioreactors, through the use of better production clones, basically. There is still a gap into what a health system can afford in terms of spendings and the costs of goods that are still involved with producing vector-based gene therapy today. So that's in a nutshell why we went for gene therapy first with our human cell design bioreactor.
So let's start about the technical aspects of your technology, Gabriel. Can you explain to our listeners how acoustophoresis works and how it can revolutionize cell manufacturing?
Yes, so you pronounced it right, acoustophoresis. So literally motion through acoustic. You have actually experienced that if you've been to a live music show. You know, when the music's very loud, high intensity, you can feel your clothes start to vibrate, maybe your body resonating with the wave itself. And so actually, the acoustic wave is, in these cases, transferring a bit of motion, a bit of energy to you, to your clothes. Doesn't shake you around because you're quite heavy versus the wave itself. But if you translate that to the cell scale, so much smaller, so you need much higher frequencies, ultrasonic, because a cell is so small and so light versus the culture medium she's floating into, then it means you can actually move quite significantly the cells with acoustic waves. So the idea is to use an acoustic wave of the same frequency that you would have when you do an echography, for instance, but to pattern it, to shape it at your will in order to move the cells you want to work with around. It can be to move them up and down because you want to separate the cells from something, from the liquid, from a contaminant, or from the thing they are producing. It can be to make them float against gravity. So that's what we are actually trying to do in our bioreactor. Instead of having the cells being stirred to counter settling, we are having very large cell culture acoustic plates, if you want, where the cells are being trapped as if they were in a petri dish or a hyperstack, but they are not resting on anything but acoustic waves. And actually, we can grow adherent cells, suspension cells like this. And so that's basically what we are doing with our acoustic technology. There are acoustic waves that we shape in order to push, to shear, or to grow mammalian cells in culture medium still, in a liquid. So it would look like a stirred tank, if you want, a conventional bioreactor, but without the impellers, without the pumps, only with ultrasonic waves inside that are performing the same functions, but that are more suited to the fragile aspects of mammalian cells.
So how different, if you look now what's going on inside of the bioreactor, how different is that environment to a traditional bioreactor? What are the moving parts and what are the stationary parts?
You have to imagine that we didn't change everything because of the specificities of the industry we talked about earlier. Basically, it is a vessel that's filled with culture medium. So with single-use components, GMP compatible. So we really try to do something that's directly applicable to the industry. Instead of having a motor that would stir the liquid with an impeller, you have speakers, if you want, ultrasonic speakers, positioned on the cell culture vessel. These speakers will generate inside the culture medium a standing wave, an ultrasonic wave, that will act on the cells. And basically, we can operate either as a batch, so you would have your liquid that stays in the vessel without inlet and outlets, or we can inject gases, as you are doing with a regular stirred tank, or operate in a perfusion mode, whether it's a pure perfusion or a recycled one. That's how we actually bring in nutrients, bring in gases to our cells. So it would look much like your conventional stirred tank without the impeller, the engine, basically, and with a different positioning of the bubbling inlets and outlets. But still, today we have prototypes that are fitting your benchtop that you connect to CO2 lines, O2 lines, to your power outlet. Our bioreactor actually consumes less electrical power than a conventional one. And you have an interface that you can use in any bioproduction lab. Basically, replaced the engine of the bioreactor with an acoustic one, if you want, but it can perform in the same fashion with the same steps as a regular one.
So compared to a traditional stir tank bioreactor, what are the advantages of your technology and what are the drawbacks?
The advantages is that you apply much less shear on the cells you are growing. Basically, you don't have anymore the spinning motion and the impeller influence on the cells. Hence, because the cells are being trapped into these very periodically spaced cell layers, they are surrounded by their peers. They are in a liquid that's almost at rest, so they are experiencing zero shear, which makes for better cell productivity and which makes for much higher cell density that we can reach. So basically, we can have at least 10 times more cells per milliliter than a conventional stirred tank, and this holds when we are scaling up. That's the main advantage today. It's also interesting to note that this approach is cell-agnostic, so it works with adherent cell lines or suspension cell lines. So it's quite interesting if you want to do process development and switch from one cell to another. You can actually do that pretty quickly and easily. And we can perform in one place, like in the liquid volume, every step that we want to do when you do like vector production, for instance, the amplification, the cell modification, so the transfection, and the harvest, the lysis, etc. So it's a tech that's also versatile. The drawbacks is that today we are still small scale. So we are operating between 100 and 200 milliliters today. Even if we have more cells per milliliter, it's still small scale. Physically speaking, it could be scaled up to 200 liters, but we are a very early company. So I have no doubts we will reach it one day. But today, if you want to get your hands on a Kolibri bioreactor, it will be a development scale, a small one. And basically, you need to be careful of what happens to your cells. It's still a new tech. We have de-risked it on HEC 293, whether it's adherent ones, suspension ones. I've worked in the past with other cell lines. But if, say, tomorrow we want to work with a new cell, then we will need to spend a bit of time finding what are the right parameters for these cells and what are the wrong ones. Wrong ones, which are the acoustic parameters that will either kill the cells, impede their productivity because the volume is too high, for instance. So basically, a tech that's improving a lot the situation, but that needs, of course, to be studied, developed, brought to a higher tieral level, if you want.
Now, speaking of scale up, yes, you mentioned that you're still very early on in development, but if we look, let's say, on a more theoretical basis, where do you see the challenges with this new technology when scaling up compared to a stirred-tank bioreactor? Because even with the stirred-tank bioreactor, it's quite an endeavor to scale it up well.
So I would say the most interesting thing about this tech for the scale-up is the fact that the microenvironment of the cells is the same when you scale up. What I mean by that is that if you see a picture of our tech on our website or online, we'll see if we can attach one to this podcast, why not? But the cells are located into very stable and periodically spaced layers in the liquid in 3D. And this microenvironment will be the same when you scale up to 1 liter, 10 liter. You will just have more layers, basically. But the cells will see the same environment. When you scale up a stirred tank, it's not the same situation at all because you need to scale the stirring rate because of the fluid mechanics equation that rule the stirring of the tank. The larger the tank, the higher the shear on the cells. Their microenvironment is changing with the scale-up. And so that's why when you scale up a stirred tank, you see quite a constant cell density if you do your job correctly. But usually you see a decrease in cell productivity when you reach thousands of liters. So the large scale compensates for that, but it comes from the fact that the microenvironment is changing. So the huge advantage of our tech is that you will have the same microenvironment as you scale up. The drawbacks are the same as for the stirred tank scaling. We need to show that all the cells have access to nutrients, gases, and that the whole volume is homogeneous, meaning are the cells on top exposed to the same temperature, nutrient, gases conditions as the same as the cells on the bottom. And basically, how are we physically doing the scale-up? Today, as I stated, we have hundreds of milliliter acoustic scales. If, say, we want to do a 10 liter tomorrow, are we making larger acoustic fields? Are we making more numerous acoustic fields but parallel one to each other? Are we doing a scale out because we have already maxed for one reason or the other the capacity of the vessel we are using and we need to put 10 vessels in parallel, for instance? That's the challenges that we are evaluating today. Fortunately, we have experts on our side that have already studied this kind of things with algae, yeast, and mammalian cells. So we are guided by experts of the field.
Hey, smart biotech scientists. I hope you've enjoyed today's conversation with Gabriel Dumy, who is the co-founder and CTO of Kolibri. Coming up in part 2, we are continuing our exploration of the use of acoustic waves to revolutionize cell manufacturing. And maybe you have a great breakthrough technology too, but you're wondering how you can make this a reality. So I want to offer you a very practical step. In the show notes below, you find a link. So I want to offer you a free 30-minute consultation to help you get started. So book your call now. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.